Thanks, Yuri. It's a great pleasure to be here. Thanks, Ferhat, Hatice, and Vishnu for inviting me. So I'm going to show you a little bit what we do in the lab in trying to build models of the immune system. And this is, of course, a long-standing interest of the group, and a lot of you guys are interested in that, obviously, if you are here. But also, I want to show you the kind of data we can generate, the kind of challenge we have in terms of doing better computational biology, because I think there's a huge space which has not been really tested. We are used to single-cell RNA-seq and a lot of big data sets, like Yuri pointed out. But I think we can really push experimentalists to do a different type of experiment to really build better models and understand immunology in a better way. So I would quip that immunology is too serious to be left to immunologists, so we need computational biologists to come into the field. So my lab is interested in a very basic process of immune response, right? We look at T cells, and we are interested in understanding how they really discriminate between self and non-self. So at some level, this is really something very fundamental that you see in very different tissues, in very different contexts. And when you're trying to understand how T cells fight cancers, when you look at the response to infection. And what's very striking at the biophysical level is that we know that the difference between an antigen which triggers an immune response and all the self-antigens is typically a single mutation. And not only that, but we know that a single peptide is enough to trigger T cells. So this is a huge field, of course, in immunology, where people have been doing some biochemistry, some biophysics, some modeling to try to understand that. And typically, you're going to get these kind of models, right? So these kind of big cartoons where people try to relate. Is there a pointer somewhere, or just like that? Okay. Try to relate the way the receptors engage the receptor, its ligands, and really get the signaling cascade going down. And this is very combinatorially very complex and very difficult to model at some level, but of course very interesting because it's really the way T cells are in a way looking at the world. And I will start with something which is a bit to grab your attention in terms of machine learning, because there is some sense that in a way, T cells are kind of like a liquid brain. We're dealing with reactions where the T cells interact with tumors or with infected tissues. And if you look at different output, in this case, I'm showing you some experimental data from almost 20 years ago, almost 30 years ago, where people are looking at dose response for a given peptide. And if you look at different cytokines like IL-2, IL-3, or interferon gamma, you see that each readout seems to correspond to a different set point in this dose response. So in a way, you can see all these different molecules which are being produced by T cells upon activation at kind of an output of the output of in a way, a neuron, if you want the analogy, which reads some different aspect of a signal. So we have been doing a lot of modeling on that stuff. At some level, we understand it relates to ligand-receptor interactions, triggering a staggered phosphorylation cascade inside the TCR. And what I want to point out for this crowd is that, again, we know that all these different stages of activation of the TCR seems to connect to different readouts. Maybe the early events would produce interferon gamma, intermediate events would produce IL-2, late events produce IL-3, but you see that in a way you have different stages of activation of TCR, and they seem to relate to how deep you can go into your signaling cascade. So you are kind of projecting the ligand-receptor interaction that you are trying to detect at the biophysical level early on into different states and different pattern of activation inside the TCR. So at some level, what we want to do in terms of modeling is to say that there is a very complex TCR signalosome, right? It's a very complex machinery, but you can use this complexity to try to map out all the different features that you might have inside your ligand, in particular different quality, but maybe other features that we have not been able to study before. So again, at some level, for some kind of analogy, what really drove a lot of what we've been doing in the last 3-4 years is that we are really using some inspiration from machine learning, where you are using some kind of antigen recognition by a TCR, which is the receptors, right? So it's really kind of one receptor in the middle, kind of a signaling bottleneck, which projects into different signaling cascade, which project into different transcriptional response. So there is this idea that you are really like kind of building a bottleneck in terms of signaling and projecting into different readout. And this architecture, of course, is really inspired by autoencoder, and we're going to really use this to build the models. So this is nice. This is kind of nice conceptually. I mean, it has been driving a lot of what we do at the theoretical level. But if you really want to train a model to be able to do that, obviously you need tons of data, right? And not only tons of data, but we know in the context of the immune system is that we can deal with a lot of different readouts in terms of cytokines, a lot of different readouts of signaling, lots of different peptides being presented. So you want really to capture the combinatorial complexity of this whole system. So to do that, we built a pipeline, what Yuri was mentioning in terms of robotics, which really enables you to do things at scale. So at some level, it's not really rocket science, right? We are just automatizing what we used to do in the lab at the bench. But again, it's a game changer because it's really going to build the kind of big data sets that we need to be able to really do some machine learning and some good modeling. So you start from the typically biological materials you can collect from clinical samples from patients or from mouse models. And then you mix T cells, B cells, macrophages, tumors, and all the different things you think are important in terms of modeling your immune response inside the dish. So it's really an artificial in vivo model, and you can start to make it as realistic as possible by using matrix or different stuff. And the key stuff is that instead of using this set of conditions and measuring a single time point and having a single readout, you put it inside a robot, which is going to go in there and automatically collect cells and process them, automatically collect supernatant and process them. And then at the end, what you end up with is a fairly large data sets where, so it's not as deep as RNA-seq, right? We are only going to measure maybe like 60 to 80 features. Typically, we measure 40 cytokines. We do a lot of single cell measurement by a spectral flow cytometry where we measure 35 markers. And again, we are going to get a lot of time point because we're in a robot. But what's very critical is that we can really collect a lot of cells. So typically in 1 week, I would say in the lab, we get 1 billion cells with these 60 to 80 features. So it's limited in terms of number of features. At the same time, we know a lot about immunology, so we can really handpick them in such a way that we can really collect things which we think are very informative. So what we get at the end is that we could do like completely unsupervised stuff. So this is just a UMAP to look at the single cell measurements, looking at the time dynamics of the T cells here, and the time dynamics of the B cells, which are presenting the antigen. So you see that it can be very rich. It's much more diverse than we would expect, something that we know from Yuri's work and others, right? And we want to really understand all these different states of activation of the T cells. But being able to map these things and trying to understand and build a simple model to capture this whole complexity is really what we're after. So if you come to the lab, what you will see is this kind of robotic platform working automatically. So again, it works by itself. I could show you something live. I'm not sure time point is being taken, so it will be a bit of a risky proposition. But everything you would do in the lab in the usual way, the robot can do, and you can program it to do that automatically. And then we have a whole pipeline on the right, which enables you to collect all the data. So I'm just showing that to give you a sense about the size of the data set that we measure. So typically, we are going to get maybe 24 times 96 conditions, which correspond to different time points, different type of T cells, different number, quality of antigen, and so on and so forth. And you can really capture all these parameters very quickly, and you end up with a huge data frame, again, with billions of cells, 60 to 80 features, and you have to build a model on that. So this is kind of published. You can refer to things. And the key stuff of what I want to show you today is that if you do plain unsupervised learning, there's no rhyme or reason to this data. So here, this is a training set that we acquired with mouse T cells. We expose them to very well-known antigens of different strength, different quantity of antigens, different time point. So this is the y-axis. On the x-axis, I'm showing you different features that we measure, like different cytokines, different type of T cell activation, and you see that there is no real good organization there. There is a lot of correlation among all these features, but there is no good way to organize them on a piece of paper because we are completely unsupervised in this case. So then the analogy I make is that in a sense, we don't have the right set of variables, right? We don't have the right type of model to look at the data. So in a sense, I would say a lot of what we do in immunology right now looks like astrophysics before Copernicus. You have the wrong model, that's what the solar system looks like, you are in trouble, right? And of course, if you have the right model and you put the Earth in the right place and the sun in the middle of the universe, suddenly you have the right set of variable, the right set of equations. And suddenly, if you want to fly your rockets to the moon, you have a better chance to get to the right place, right? So I think a lot of immunology today is really in that state where we are looking at many different patterns and we are trying to build some geometry in some way to organize the data, but because we don't really know how to organize the data and we don't have the right set of equations, things look arbitrarily complex. I mean, it can be very pleasing to do geometry in this context, but way too complicated. And of course, if we want to move to clinical applications, we'd rather have a model like that to be able to fly to the moon, right? So there is a whole field of machine learning, which is kind of using autoencoders to do this kind of stuff. So you take all these parameters. So this is a paper from 2020, where the group in Switzerland picked up all these artificial data, which are astrophysical observations, fed them into an autoencoder, and tried to really collapse the data to build a key model. So you feed all the angles between all the different planets as you would measure them by astrophysics. And what you learned is that the best way to capture the complexity of data and to have a low-dimensional representation is really to put the sun in the middle of the universe. And in a way, you can do Copernicus in a computer in 10 minutes very quickly. So we are trying to do kind of the same thing on our scale with our immune data. So again, if you look at the raw data, so I'm just showing you our first pass with these data sets where we are only looking at cytokines. So I cannot show you a 40-dimensional space, but I'm showing you the three most informative cytokines, so interferon gamma, IL-2, and TNF, and it's color-coded according to a different quality of antigens. And you see that, again, it's very nonlinear, it's a bit complex, there's a lot of overlap, and you don't have a very good separations based on the different type of antigens activating the cells. Okay, I can skip that. So what we did is build a very simple machine learning. So we tried a lot of things at first, and this is really a collaboration with Paul François at Université de Montréal. Very complex architectures. You can really capture the complexity of the system, but you cannot really predict anything. What we wanted is really to build a model which gives us a chance to really make predictions and move forward in terms of immunology. So what we ended up with is a very, very simple architecture, right? So multi-layer perceptron, but with a single layer at the end. Things are getting more complex now that we do single cell modeling, but in the context of this cytokine, we take the raw data, right? Again, time kinetics of like 40 cytokines, a function of time. We pre-process them by really doing the time integral of the log, and we can discuss that if you're interested in that. But there's a lot of feature selections, which is really based on the way we understand the biology. So in this case, integral of the log of cytokine is really the best way to feed our neuronal network. And then we are really trying to collapse it into a low-dimensional latent space according to the different quality of peptides that we have. So again, the task at hand is really to try to build a model. We are kind of reverse engineering the way T cells are recognizing these different antigens. We are stating all these different cytokines are kind of output and features which are being produced by the T cells. They must contain a lot of information the way the T cells are activated. Can we reverse engineer it and really build a low-dimensional space, a low-dimensional latent space, which really captures all this data? And again, at some point, it's set as a classification problem, but I'm going to show you that it really enables us to build a continuous model and really learn some immunology. So what we ended up with is a latent space which looks like that. So this is, again, color-coded for different quality of antigens. So you see that from the raw data, which were very nonlinear and very mixed based on the antigen, we can really build a very nice classification where all the good antigens in blue seems to be moving kind of together. All the intermediate antigens in yellow and green are in between, and all the bad antigens in purple are practically in a different place. So in the low-dimensional space, we have all these dynamics of the latent variable, and from there, we can really reconstruct all the data. So again, I'm just showing you the most critical cytokines, but from a 40-dimensional space, we collapsed it into a 2-dimensional space with fairly simple dynamics, as I'm going to show you. And from there, we can really rebuild all the data. So in a sense, we are making progress because we went from a very high dimensional space to maybe a 2D space. And in terms of reconstruction, I will stress that this is a huge range of measurements. So cytokines really accumulate. So we can really go from 100 femtomolar to practically 10 nanomolar. So we have practically a 10 to the 5 fold range of cytokines. And this very simple encoding, this very simple latent space seems to be able to collapse all the data and really rebuild it all the way from the very rare cytokines to the very abundant cytokines, early time point, late time point. We really capture everything very easily. So I think we are making progress. Again, not Copernicus yet, but at some level we are making progress because it means that from a very complex set of features and a very rich time dynamics, we have a low-dimensional representation where maybe now we can look at things in a better way and really try to understand the way T cells are wired. Sorry, Yuri, 40 minutes is with questions, or you have time for? Okay, so I will try to. Yeah, so then we did so. I'm a physicist by training, so being able to reconstruct the data is a nice thing, but we really wanted to analyze this latent space. We really wanted to understand what it means in terms of immunology. We really wanted to look at the guts of this model to really understand what's going on. So in this case, it's fairly simple. So in discussion with Paul, we ended up parameterizing the latent space with 4 variables. So essentially, all these trajectories are going to correspond to initial speed with different angles, which correspond to the ratio of adaptive and innate cytokines. The system keeps speeding up to T0, which is the time when it starts to fall down, and then there's a final speed. And this is a kind of fairly complex model to capture the nonlinearity in the system. But from this 4-dimensional space, in fact, we show that when we fitted our data, all these variables are practically collinear, except the last variable, but it's not very informative. The speed, the angle, and the time of accelerations are all proportional. So at the end, we end up with practically a 1-dimensional parameterization of all the dynamics. So again, big progress, because we started with a 40-dimensional space of cytokines, collapsed it into a 2D space, latent space, and now we can parameterize the dynamics inside the latent space with a single variable, which is this immunological speed. So I think we're making progress. We can do some math to really understand a bit some information content. But what's very important for us is that we can go back to this variable in this latent space to really understand the way T cells really discriminate between different antigens. So what I'm showing you here is like a Poincaré map at 36 hours. We cut the latent space and see where the system was at 36 hours. And we plot it as a function of different quality of antigens on the x-axis. So again, I'm showing you the latent variable 1 in yellow and red for latent space variable 2 as a function of the different quality of antigens. And here we are really going to learn something that we can use to go back to the lab and do some biochemistry. So what we learn is that if we look at the yellow variable, there's a very continuous dose response. So the better the antigens, the more you activate this mode, right? And more interestingly, when you look at the second variable, so this variable contains practically 80% of the dynamics, and the red variable adds practically 18% to really capture almost all the dynamics. But the interesting part is that this red variable is very nonlinear. What it's telling us is that if you look at the self-peptide, the self-peptide doesn't activate either of these modes. You don't have any kind of motion inside the latent space. Very good antigens, which is what you would expect if you're dealing with a virus, when you're dealing with a very antigenic tumor, would be here, where you activate these two modes in a very positive manner. And that corresponds to a very full blast activation of the cytokines. But more interestingly, when you're trying to be in the intermediate regime, when you're looking at the antigens which are kind of in between, you don't activate much the system in terms of the first variable, but you activate the second variable in a negative way. What that means is that there is a negative imbalance between adaptive and innate cytokines, which seems to be the hallmark of all these peptides which are in between. And this is very critical because, again, we wish that most tumor neoantigens are here, but in fact, practically most tumor neoantigens are very weak activators. And what they do is that, if anything, they tend to activate negative feedbacks. That's most likely why tumors can escape an immune response. So we have, again, from our modeling, from the latent space, we have a very easy way to understand this regime, and we are going to be able to redo biochemistry to modify it and maybe make better T cells. So this is just purely about T cells. And again, this was published 2 years ago. So I refer you to this paper to look at all the details if you want to look at the gory details of machine learning. But what I want to present today is things which are a bit newer, which relate to CAR T cells. So CAR T cells are a whole new slew of biological products that are being used in the clinic to fight tumors. So it's inspired by the design of normal T cells, right? You have the receptors, you have signaling cascade, and then usually you have a costimulatory domain. And then researchers, first at Weizmann in the 90s and more recently at Sloan Kettering and at UPenn, and then now everywhere, are building this construct where you are using an antibody as kind of a receptors, right? So a single chain antibody, which is going to be the extracellular domain. And the intracellular domain is going to contain some of its costimulatory domain and some of its TCR domain to really build a chimera, so a chimeric antigen receptors, which is able to recognize the tumors based on the antibody and able to signal to the T cells based on the signaling domain. So this is kind of a mishmash of many different things. And at some level, there was no real quantitative models to look at the way these cells are activated. So we took these cells in collaboration with Naomi Taylor and Nihal Isha at the NIH, and we tested, again, we trained our models in normal T cells at the top, and then we took these CAR T cells and then tested the way they really recognize tumors. So again, we end up with a latent space in 2D where you can really rank the different antigens based on their quality, and this is really based on what we have learned from normal T cells. And then in black, I'm showing you where the CAR T cells lines up compared to all these different ligands. So it's a bit abstract, so I'm simplifying it with, again, our immune velocity and our parameterization. For different quality of receptors engaging antigens, you get a nice ranking of it about the speed of activation in the latent space, right? So the self is not giving you any kind of activation, there is zero speed. And when you have very good antigens, you get here. And when you take the CAR T cells, and everybody in the field believes that because CAR T cells are made in a kind of optimal manner, you're going to get something which is as good as it gets in terms of T cell activation. In fact, not at all. When we compare CAR T cells to normal T cells, in fact, they are almost the crappiest T cells you could expect. They are kind of activated in the crappiest manner when you look at the latent space. So again, this is a model which is really trying to classify immune activation based on different antigens. So maybe CAR T cells are very good at killing, but what we are stating is that if you are trying to compare them to normal T cells and normal trajectories of immune activation, you are in trouble because in a way they line up with the worst antigens that you could have. Not only that, but something I forgot to mention when I was discussing the negative feedback, right? So this means that there is a negative feedback being activated for the intermediate peptide. And when you have a negative feedback, you expect antagonism. You expect that if you have two signals, one good and one bad, the bad one might activate so much negative signal that you're going to end up antagonizing the activation of the strong one. So I'm pointing that out because when we look at the combination of the CAR signal on the right and the TCR signal, so again, we repeated the experiment where these T cells have a TCR which recognize peptide with different quality. So that's what you see on the x-axis. And they also have a CAR which recognizes the leukemia with CD19. And what you see is that in some conditions, you do even worse than the CAR alone, right? So mixing two receptors, one CAR, one TCR. And if you engage the TCR with a very weak ligand, which is like the self, it seems that you're in this regime where you get the negative feedback and you are really going to block CAR activation. So you really degrade the activation of a signal. And vice versa, if you have a very good ligand and the CAR signal, you do better than the CAR alone. So you see that things are not linear, right? There is a non-linear combinations that we have to really tackle if we really want to understand how CAR T cells are wired and maybe make better CAR T cells. So that's what I'm going to show you in the next part. So the first thing we did to really convince the clinician that it was something very important to follow is to have a little bit of an in vivo model. So again, we build CAR T cells. This is our training set where we have a CAR T cell which contains this CAR which recognizes CD19 on leukemia. We have a TCR, which is our OT1 training set, which recognizes ligands of different strength, right? And we're going to be able to test different tumors. Some of tumors are going to present no ligands for the TCR. Some tumors are going to present some very strong antigens, and some tumors are going to present some intermediate antigens. And here I could make a survey and ask you what you expect, right? But no, if you take, so again, the experiment is fairly simple. That's a classical assay that clinicians do when they want to validate a new CAR design. You inject the leukemia with a CAR target. In our case, the leukemia also expresses an antigen for the TCR. And you see in green that you measure how long the mouse can survive because the leukemia is growing. And if you have a CAR injections, hopefully they can delay death compared to a mouse which is just exposed to leukemia. So in green, you see that the mouse survives a little bit more. If it was a tumor by itself, it would be like 20 days, and maybe it can survive 50 days with a CAR treatment. If you have a tumor which expresses both a CAR ligand and a very strong TCR signal, you can fully protect the mouse, so you can really eradicate all the tumors. And if you have a weak intermediate ligand, maybe you will expect something in between. In fact, not at all. what you get is that, in fact, mixing a CAR signal in a weak ligand, you make practically a CAR completely obsolete. It doesn't work at all. The weak signal which goes through the TCR is really completely antagonizing the CAR activation. You end up with the worst clinical product in the world that you would expect, because it does as bad as not doing anything at all. So again, this is really nonlinear, right? From no ligands in green to weak ligands, which gives you a negative signal, to strong ligands, which gives you a positive signal, things are very nonlinear, and we needed a way to really be able to model that in a smart way. So to make the math very simple, sorry, usually I present this slide for biologists, but for this crowd, it's going to be a bit offensive, but it's okay. When you mix, again, CAR and no TCR signal, maybe you have one signal. You would expect that CAR plus strong TCR is 1 + 1, maybe you would get two signals, and CAR plus a weak TCR is 1.2. But in fact, not at all. When you mix, so the anticipated result would be a linear combination, and what we see is that it's very nonlinear. A CAR plus a weak TCR is something less than the CAR with no TCR signal, and we wanted to model that. So again, we could have gone and do a lot of biochemistry and do the classical work, but because we had our machine learning tools, which enables us to really collapse all the data and really test these things very efficiently, we went very quickly to a very good model to be able to do these models. So we went back to our robot and did a lot of data. So again, big carpets of data where we test different CAR targets, different quality of ligands on the TCR, different ratio of cells, different type of tumors. So we tried to really cover the space of all the possibilities in terms of this mix of TCR signal and CAR signal. We built these big data sets. And here we went back to a very simple biochemical model, which is a bit more like what we are doing pre-2018. So we really do like some MCMC, and this is really done by Paul's group again. We can really go and parameterize this model in such a way we can really capture this whole system and make good predictions. So practically, what does it look like? You have a model where you have two signals going through the pathway. So one which goes through the TCR, one which goes through the CAR. And the important idea, which is again something that we have analyzed from our latent space in the first part, we know that there is a shared negative feedback between these two pathways. So as you engage this TCR with a weak ligand, you're going to activate this negative feedback, which is a SHIP1 phosphatase, which is going to be a crosstalk between the TCR and the CAR. In that way, you're going to be able to explain some antagonism, and you're going to be able to explain the complex math that these CAR T cells are able to do. So we had a classical model where the negative feedback was being activated as we go along and really impinge on the progression of this signaling cascade. And we did some MCMC to be able to optimize the parameters. And we ended up with a very crappy model with very weird posterior distributions, where in a sense, we didn't have a good fit of all the data because we had so many conditions. We were not able to fit all the conditions across with one set of parameters. So we went back to the drawing board. We made a new updated model where now the negative feedback is activated very upstream in the signaling cascade, but it only acts on the very downstream signaling element. So again, there is some biochemistry justifying that, but this comes really from the way the machine learning is being designed. So we always check that these biochemical models are compatible with the latent space that we measure. And we do, again, some MCMC parameterization. And now we end up with a very well-defined model with one given set of parameters where we can really account for the nonlinear interactions between the TCR and the CAR. So the results are here, and the fit is a continuous line. What I'm showing you here on the x-axis, different quality of ligands, TCR antagonist on the x-axis. On the y-axis, I'm measuring the response of the CAR plus TCR system compared to CAR alone. So again, if you are exposing the T cells to a self, you have on the CAR only, so it's 1. If you're exposing the T cells to weak ligands, you get antagonism, so you go lower than the CAR alone. And if you have a good ligands, maybe you go above 1, you get some enhancement. And you see that the model with like practically 4 parameters is able to fit all these conditions of different quantity, different quality of ligands in a very systematic manner. So we have progressed in the sense that now we have a universal curve which tells us if you give me a TCR, if I can measure the quality of a TCR peptide MHC interactions, I'm going to be able to predict in the context of the CAR clinical trial whether this is going to enhance your therapy or otherwise going to antagonize your therapy, and you have this negative effect happening. So we can make it work in human cells. I'm going to skip that part. But really, the message I want to give you, and something which is maybe more general, is that there is a whole space of understanding immunology which has not been explored enough, which is that many people are trying to build models of the quality of antigen that we have. So typically what we do in the clinic is that we harvest tumors from the patient, we sequence the tumors, we use a different model of antigen presentation to try to predict what type of antigen exists. Very often we don't really have good model to be able to estimate the antigen strength, so it's a big challenge for the community. But not only that, what I'm positing is that we have to pay attention not only to the neoantigens, but we have to pay attention also to the wild type counterpart. Because again, right, you are looking at tumors, you are looking at a neoantigen, which is a mutated bits of a protein, which is really driving, hopefully, oncogenesis. But there is always a wild type counterpart, which is one mutation away. And my point is that because we know about antagonism and we know about this nonlinear effect happening in the T cell activation, we have to pay attention that for given antigens, the wild type counterpart can be an antagonist. And if you look at another neoantigen which might be weaker, maybe the wild type counterpart is going to be a real self which does nothing, right? So in terms of modeling, we want to be in a regime where we can really line up all these numbers based on the way we measure T cell activations. And I think it really maps into different state of activation of the T cells and really understand our combination of neoantigens and wild type ligands give you different effects. So this is the kind of modeling that we are doing. Typically, experimentally, we start, and these are data from Marta Lushka's group, Vinod Balachadran, at Sloan Kettering, in the pancreatic vaccine clinical trial. They tested different TCR of which they knew the antigen on the x-axis. So this is a sequence of the agonistic peptide. And they provided all the single point mutations. And these are all these EC50, all the degree of activation that you have. So it's a very complex matrix. In that paper in 2022, they proposed the model. We are proposing a different model now. But what we are stating is that depending on the strength of all these different mutants, we can predict how much antagonism or how much enhancement we're going to get, right? So it's going to be extremely nonlinear. And it varies a lot depending on the type of TCR that we have. Some TCR, every mutant that you're going to make are going to enhance the response. Some TCR, every mutant that you make are going to be antagonist. And you have all these things which are intermediate. So again, based on the modeling and based on the machine learning, we need to be able to predict what class of TCR, what class of immune activation we are dealing with, because we really want to be able to predict what the self is going to do in that context, to be able to modulate the immune response. And again, we can discuss more that in more details, but there is a whole space of machine learning which needs to be done to start from the sequences of the TCR and the peptide MHC to really try to predict how these things are being set up. And it's very devilish because small differences really imply huge differences in terms of immune activation. So I'm going to finish with some practical consequences just to show you that it's not purely a modeling effort. So classical CAR T cells, for example, the CD19 CAR, which is used against leukemia, kills very well the leukemia, but it kills also all the B cells inside the patient. So what you end up with is a patient for whom hopefully you have cured of all the leukemia, but at the same time you induce B cell aplasia because you have killed all the healthy B cells. There is no way to discriminate between tumor and healthy B cells. And it's not a big deal because, again, B cell aplasia, you can compensate with the right treatment, and these patients can live for a very long time without their B cell compartment. Now, if you try to use the same idea of designing a CAR with an antibody which recognizes a solid tumor, you can, again, make a CAR which is very good at killing the tumors, but very often they kind of have some side effect. And this is extremely problematic because you can live without B cells, but you cannot live without your lungs, right? So every time there have been some clinical trial with CARs targeting solid tumors, you get good results in terms of killing of the tumors, but very often you end up with a fatal toxicity because you are not able to discriminate between these different ligands. So again, we can do some modeling, but for the sake of this discussion, I want to show you that what we learned from the model in terms of this antagonism, we can use to design a better CAR, because if you make a CAR where you control not only the CAR, but also the TCR, you can pick a TCR which is going to be able to recognize the tumors as the neoantigens, and this is going to be enhancing the response and giving you a positive effect. And if you pick the right TCR, it's going to recognize the self as an antagonist, and it's going to really block the activation of the CAR. In that sense, you're going to protect the healthy tissues. And again, thanks to our models, we are really able to pick and really fast track the way we pick the right pairs of CAR and TCR. And indeed, when we go to an in vivo model, we get into a regime. So this is an in vivo model where we are comparing the killing of healthy cells in the lung measured by fluorescence and the killing of the tumors on the flank. And we end up with a perfect regime where we can really kill the tumor very efficiently. And it's not perfect, but it's much better than what you had before because we don't kill the healthy tissues in the same samples. We are kind of getting the right space. Sorry, the end was a bit fast, but I hope it gave you a flavor of what you can do. I wanted again to give you a sense that we can really build big data sets. So it's not just for us. We really hope that the community can use this kind of data, and we get tools from you guys to really tackle this data in a better way. It's fairly easy for us again to get billions of cells with a lot of features, and more importantly, with a time resolution to be able to retrack the way T cells, B cells, and NK cells get activated. This robotics is fairly easy to implement and fairly cheap, so we hope to see it in many other labs. From a machine learning point of view, being able to compress the data in a realistic manner in such a way that it really lines up with biochemistry is a really critical stuff. It was super useful for us to really build a model of antigenicity and really learn the way T cell discriminate between self and non-self. And again, identify the negative feedback, which seems to be critical for that. And again, when we move to CAR T cells, based on the model, based on these big data sets, we really can learn about antagonism between TCR and CAR, and really be in a regime we can really build better CARs, which can really protect the healthy tissues. So this is the lab 6 months ago. So we are a small group, but we have a robot, so we can do a lot of experiment. And a collaboration with Paul Francois for machine learning at University of Montreal, and a lot of help from Naomi Taylor as well for the clinical side. And looking forward to your questions.
May I ask you?
So I have two questions. One is for the T cell modeling, the first part, because we know that in order for T cells to activate, we need antigen presenting cells. And in your model, you have a lot of parameters in terms of like antigen concentration and the time points and different readout. However, I didn't see that the MHC is highly polymorphic. So do you take into that account in your model?
Okay, so the training set is made with a very well-defined system where we have a clonal population of TCR, so we know exactly it's fixed, right? And we are dealing with a very well-defined peptide MHC, so very well controlled. You're absolutely right that once we have parameterized this, we can move into a polyclonal population with a whole repertoire of peptide MHC. Indeed, that's what we do. You also mentioned the AP, the antigen presenting cells. Here, I showed you only a model about antigen recognition, right? So we are doing abstraction of whether it's the dendritic cells, the tumors, and so on. We only want to build a model about the way the TCR interact with the peptide MHC. And indeed, we could do that, right? In a way, it's surprising because if you look at one readout, it's going to be all over the place, depending if you have the right inflammation or the right signal. And yet, because you have a high dimensional space, you can compress it in such a way that it lines up with the TCR quality. And you can do a distributions. You can go into a regime where it's polyclonal, where it's not controlled. You have a good model to line up the data according to that.
Okay. All right. Thank you. And the other question is not directly related to your T cell, but about right now a lot of therapy talking about BiTEs, bispecific antibody therapy. Are you considering building similar models?
Correct. So we do have, in fact, the data. We're collaborating with Mike Dustin at Oxford, who is doing a lot of work on BiTEs. And you can do exactly the same game, right? So you use BiTE, and you do it in many different settings, different affinities of receptors, different ratio, different type of tumors, and try to build a model of a signaling cascade and what comes out of the T cells, such as you can, again, collapse all this data. And it looks essentially exactly like, so human BiTE versus mouse TCR, it looks essentially the same. And in that way, you can really rank these different BiTEs according to the quality of the TCR. And we think it's going to really map out in terms of T cell activations and the type of T cells we can produce. So, yeah.
Thank you.
A quick question about the autoencoder architecture. I just wonder why 2 latent space, and is there operational reason, and have you tried like other numbers, and does it give you like more sort of resolution in your modeling?
So at the end, I'm telling you that's a 1D model, right? We only care about anti-GCT. So of course, we are really trying to push a lot of architectures to really get only a 1D model at the core because we wanted to automatically get this stuff. But somehow having 2D is good enough to really get this negative feedback and really explain this very fine resolution we were not able to, again, we are not machine learning people. So I'm sure smarter people, I hope smarter people will tell us a better architecture to do it. That's how we got to learning about the negative feedback and being able to do more experiments. 3D doesn't add anything. And I'm sure better architectures, and again, now that we have single cell data and billions of cells, we are doing 3D latent space because we are learning additional product mechanisms. But in a way, each dimension is a mechanism which maps into biochemistry. In that way, we can make some progress. So right now it's 2D because that's what's useful. I'm sure you come back from a few years from now will be 5D, where we understand each of these dimensions in terms of map kinase to NFA to different things. And in that way, we really start to build the complexity, rebuild from scratch the complexity of immune response.
Thanks for a very terrific talk. So the question is kind of similar to connect to the last one. So if you used a more traditional latent factor method like a CCPIS, would you get similar results?
Yeah, so we tried the smallest, the complexity we are capturing. So we have to, you can go back to the paper and we do some information theory to really quantify how good our latent spaces in terms of being able to capture all the information content of these antigens. And this architecture was practically capturing everything and was the best. If you do different methods, it was a bit downgraded. It's not too bad, meaning that it's not too hard to capture low dimensional space, which is fairly good at discriminating between antigens. This architecture was just the best. So that's what and that was the easiest to interpret in terms of biochemistry. That's what stuck, right? So.
Are you planning to produce similar large data set using human cells?
This is so yeah. So I showed you only the training set, which is mouse cells. Another surprising stuff is that all the parameterization we got completely applies to human cells. So typically, what we have in the labs are naive T cells straight out of the mouse. What we have from the clinic are usually T cell blasts, which have been expanded for 2 weeks, right, which have been harvested from patients. And yet, in a way, this is reassuring. The model we learned from this training set applied directly to the human cell and was very good at ranking the antigens. So I think we have a good model in hand. Yeah.
Great, thank you.
Thank you.
Can I ask, I think we'll close this now. I just have one last question. So for any of your measurements about cytokines, do you care, for example, negative feedback loops or more generally, do you care about how these molecules, cytokines, for example, signaling molecules, are actually regulated at the chromatin, at the transcript level? And a related question, what's your view? I mean, many people will approximate these with transcript levels, cytokines or signaling molecules.
Yeah.
Do you think it's a good approximation? Or?
It's terrible.
Okay.
Sorry, Yori. But in fact, cytokines is really very translationally regulated. So of course, with a big enough model, with enough chromatin and so on, you might be able to capture that. We definitely know that when you look at cytokines, because they are secreted, right? So it's not, there's a whole process of maturation of a protein and being able to be secreted where you don't capture as much dynamic range and as much things that you get from transcriptomics. From our side, it was just cheap, right? Measuring protein, we can really do all these assays, literally 1 cent per well. So we are being cheap. That's why we didn't do transcriptomics. We really hope to get, right, if there is a low dimensional space from the protein, there is of course a low dimensional space from the transcriptomics, and these two have to jive together. We're not there yet, but yeah.
Very interesting. Thanks so much again.
Thank you.